There we go. Okay. Hi, everyone. Welcome to the second session of our webinar today. We're joined by faculty members who have plenty of experience mentoring grad students and interns. So they are going to be here today to answer all of your questions. We have some really great questions that were submitted by our attendees today. So I think we'll jump right in. I'm going to start with letting them introduce themselves and also talk just a little bit about the research that they do in their lab. So I think we'll start in order and we'll go with Katrin, Mia, and then Andrew. If you wouldn't mind introducing yourself and then just really quickly talk about what kind of research you do in your lab.
Yeah, hi, I'm Katrin Kapstein. I'm sorry I already barged in before. I am at the Florida campus, and I actually run the undergraduate program there as well as the prep program. And so that's why I felt justified in being super rude. Anyways, my lab is interested in understanding how ribosomes are assembled and how that assembly is quality controlled and how defects in assembly then lead to disease in humans. We do this mostly in yeast, and so it's a way that is really amenable to undergraduate research. And we use a number of different techniques from, genetics, genomics, biochemical, and also some structural techniques.
Hi, everyone. My name is Mia Huang. I used to be an investigator in the Florida campus, and I recently moved here to the San Diego La Jolla campus. My lab studies glycans, and we are on a mission to understand what they do in the cell. Glycans are essentially carbohydrate molecules, but we don't really see them as nutritional biomolecules. We see them as recognition molecules. They interact with proteins to enact important functions in the cell, and therefore they are important targets for disease and important targets for designing therapeutics. So my lab is designing a bunch of chemical biology related techniques rooted in synthetic chemistry, but also cell biology to be able to address these questions.
Great. My name is Andrew Su. I'm a professor here at San Diego campus. My lab focuses on bioinformatics. Bioinformatics is really this intersection between biology, computer science, and statistics. And so we think about how do we look for interesting patterns in large data sets. That could be identifying which genes might be new genes that are involved in specific diseases, or identifying drugs that might be used for various diseases. And so in terms of internships, right, my lab in terms of who's in my lab, right, we have all sorts of people who come from a biology background who are looking to learn a little bit of computer science, as well as people who come from a computer science who want to apply their skills to sort of biomedical problems.
Great, thank you. So that leads me into the next question. Next topic would be, what are some examples of some of the types of projects that interns have worked on in their time in your labs? And we can go in the same order, Katrin, Mia, and Andrew. Oh, sorry, Katrin, you are muted. There we go.
Sorry about that. So, I mean, it depends really on how much previous experience the students have. We really like to tailor projects to the previous experience of the students and also a little bit to the interest. So oftentimes we do projects that start with some yeast genetic experiments, because these are very, most people can do them. We sometimes have high school students do them. And so then the questions are really oftentimes along the lines of if you break one thing and then you break a second thing, how does this change something? And so we use really, so I guess I'm not answering it in a very specific way as in a question, but this is just explaining how we use yeast. So a project, the last person who worked in my lab this summer was trying to understand the molecular basis for an observation in the literature that half of all human cancers have lost a specific modification in the ribosomes. And so we had a hypothesis for why that is. And the hypothesis was that the modification is essentially a molecular check mark on the ribosome that says this ribosome is correctly assembled. And so the lack of the modification was basically saying half of these ribosomes in half of all cancers are misassembled. And so she then used a combination of, again, yeast, genetic experiments and biochemical experiments to test that hypothesis. And it seems to be actually true. And again, and we worked with her in her previous experience to, make this happen in the lab.
I'll echo Katrine's comments on being able to tailor everyone's internships based on what their previous experience is and what their current skill sets are. The interns in my lab are typically exposed to a smaller set of questions, like in Katrine's experience, something that we found unexpectedly that we want to explore a little bit further, need a little bit more data. Now, to answer that particular question, we need someone trained in cell biology. So you need to culture mammalian cells, make sure they're alive and they're happy, knowing how to recognize how they are under the microscope. So you learn how to use a microscope as well. You learn to identify what the little different organelles are in the cell and to understand what features makes a happy cell or an unhappy cell. So that's sort of the basic. Then we teach you how to pipette and work with tools in the lab so that you can measure liquids accurately, so that you can add different types of perturbations like drugs or other type of molecules to these systems, and then understand what the corresponding effect is so that we can further explore the hypothesis. So answering a question, even if it's small, requires someone to be trained in a little bit of cell biology, a little bit of analytical chemistry, knowing how to interpret data, knowing how to prepare a PowerPoint presentation so that you can actually share your data with the group and to me so that I understand what you're doing. So it's like a it's a fully intensive experience. Immersive, sorry.
For my lab, it really comes down to whether people see their, whether they are coming in with more experience on the biology and chemistry side or more experience on the computer science side. And we use that as sort of an excuse to learn a little bit about whatever side you have less experience on. So an example for somebody who comes in with a little bit more biology experience, one example project we did recently was to look at tuberculosis and doing protein family mining in tuberculosis. So looking at sequence similarity between the proteins in tuberculosis and other pathogenic bacteria. And then for people who come in more on the computer science side, who have experience in computer science, and nowadays there are more and more like amazing high school students who have a lot of expertise in this area. There we can look at building computational tools to look at data integration or look at the pattern recognition process, sometimes organizing data essentially to do machine learning on it, or potentially some machine learning projects themselves.
Great. Kind of keeping along the same lines of topics of projects, are interns ever allowed to come up with their own projects and ideas?
I'd say it's extremely rare. I think every now and then it's something that where someone's interest is maybe taken into consideration, kind of. But I think that first of all, it is a very short time. So most of the time I build a project that already kind of similar to what Dr. Wang said. There's already an observation in the lab that is foundational, because otherwise you cannot complete anything in 10 weeks. It sounds like it's a whole summer, but really it's not very much time. And then, the expertise of the lab also has to really match. And so I think those are two strong reasons why I think it's more beneficial to you do a project that's really rooted in the experience of the lab. I think even for graduate students, it's very, very, very unusual that they come up with their own projects. It can happen maybe again what's more common is that there's a student who says, oh, I've been working in this lab and I've been working in that lab. Can we maybe do a collaborative project? So that's the most common thing that could happen at the graduate student level, I would say.
Echoing Katrine's comments again, I think for you to be able to have a really productive internship, you must be able to identify the core competencies of each lab. For example, I barely do any computer work. I cannot do bioinformatics. Making me do that will be very painful for me. And so I can only do that in collaboration with someone like Andrew's lab. Not to say that sometimes it's not impossible for new questions to arise, and you're more than welcome to talk through all of them and with us. But I think there's also a difference between something that's just interesting, something that's a question, and calling something a project that we're willing to invest in, that's worth investigating and putting people on it and resources on it. It's all a very different tier in my lab.
Absolutely. I think there's you can always do the thought experiments, right? Brainstorming ideas and thinking about how you might approach a problem. Absolutely. I think we all want students to be able to do that, but there's a lot that goes into making a project shovel ready, right? To actually make progress on that. I've had that essentially maybe once or twice where a student has come back, right, either working during the school year or things like that, where they've come up with sort of their own idea, and it was sort of something that we can actually act on. But I would agree that it is pretty rare.
Great, thank you. Okay, let's see here. We have lots of great questions. Let's move over to kind of mentorship style questions. What is your philosophy when it comes to mentoring students or interns in your lab?
I think that you wanna, well, first of all, I would say a big part is that every person is different. And the most challenging part is to figure out everyone's strengths and obviously also what is it that they are, maybe not their weaknesses, but what are the things that they feel most insecure about, because you then want to try to build on the strengths and not on accident expose their insecurities. So trying to figure this out, I think, is really key to a productive interaction in the lab. And there's unfortunately no cheat sheet, right? That's the trick. And then I think the other thing is to I'd say my philosophy is to really challenge students, because I think that, and probably I have a reputation for this, but I think in the end, then the students can really look back with pride on what they have accomplished. And so I think the expectations, my philosophy there, maybe you could also say is I have high expectations, and in my experience, students tend to rise to those expectations.
My philosophy is also quite similar. You need to be able to communicate with me about what you want to get out of this internship. If you're going to tell me and say, I want to go to med school and I would like to gain some, how to think about science and how to think about a scientific project, then that's really helpful for me so that I can mentor you to help you think about it like that. On the other hand, if you are also saying, I'm not quite sure about a career in science, I'm using this internship to tell me whether this would be a career path for me. That's also really helpful. Please don't feel afraid to let your mentor know that that's sort of what you're considering. We can talk you through the pros and cons about what graduate school might be like. But mentorship is very individualized, but it does require some work from you as well to be able to communicate your needs to your mentor.
I don't have anything more to add. I think it is a very individual thing. So tailoring that to the individual as much as possible is, I think, key.
Great. So then that kind of leads into this next question, is that you are all great supporters of educational outreach and internship programs. Why do you think it's important to support these interns and students?
I mean, to answer the specific question that you're saying, why is it important, is because we want to still do science in 5 years or in 10 years. And those, in order to do this, we need to train people. A slightly different but related question is, why do we, there's all kinds of stuff that's important and we don't necessarily do them all. Why do I like to do it? Why is it important to me, and why do I spend my time on it? Is because I think it is actually the most satisfying thing because the growth that the students display, especially that early in their career, is really unbelievable. They come into the lab not knowing anything, having ever worked in a lab, and they leave with having discovered something that no one else in the entire world knows. It's just, them and me. And so I think, to see that excitement and how they, develop this pride in their research, it's really it's just super fun. And so that's, it's not exactly the answer to your question, but yeah.
Same reasons for me: feeding the talent pipeline, because you guys are going to be the future scientists, paying it forward. Someone did it for me, so I want to do the same. And then selfishly, man, there's so much energy that interns bring into the lab. There's like a lot of chatter, a lot of questions. I really want the interns to be asking a lot of random questions sometimes, because it stimulates discussion, because that's something we had never thought about. So in return, you're feeding us energy that sometimes we're just like really sad about experiments, and that kind of enthusiasm just is really re-energizing. That's why I like having interns in the lab.
It's tough going third. I think Katrina and Mia hit it all. I mean, I think I have nothing to add. Perfect.
Great. Well, I know Katrin said something really interesting about how, it was really exciting as an intern or student to discover something that no one else has discovered. And that leads to another question, which was, how have interns contributed to discoveries that have been made in your labs?
Andrew, you can go first. Although this one, it doesn't matter at all.
It's fine. I'll go first. Yeah. I've been really lucky to have some great interns, right? I mean, internship experience runs the gamut of things that are very, very, very focused on the learning experience for the intern. To where the intern comes in with a lot of experience, and really it's the excitement of applying those existing skills to exciting new projects. And so we run the gamut. I had a high school intern who essentially came in with a ton of skills and really had great contributions to a grant project we had, again, all about data integration. And so he ended up continuing to work remotely. One of the beautiful things about bioinformatics is that remote work is perfect. So he stayed with us into his freshman year of college and through 2 summers, and that's been a fantastic experience, I think, for us both.
For us, you're an extra thinking brain, an extra pair of hands. The past 2 interns in my lab have published papers with us because after their first week or so getting trained and understanding what the question we're trying to solve is, we need another person helping us do these experiments and repetitions, and an extra brain thinking about are there additional factors that we should be considering in conducting this experiment. So all of that contributions earned them an authorship in publications in our lab.
Yeah. And that's true for me as well. I mean, I already told you about the last student. Remember the hypothesis about the ribosome modifications. The specific hypothesis was that it changed an equilibrium between 2 different structures. She acquired data for this, and we still are cleaning it up, but she will become an author on a paper. I've had summer undergraduates become authors on papers because they've oftentimes they provided initial data, either they provided initial data to support further investigation, or they they made a figure that will become part of a paper. So like once we had a student, I had a postdoc who was working on roles of these factors involved in ribosome assembly, and this involved a subcomplex. So I had a student mapping protein-protein interactions, making a bunch of different truncation mutants. She was an author on a paper. We had high school students that became authors on papers, sometimes for scoring phenotypes. So I mean, essentially, this, like Mia says, the students have hands, and after they're trained and they can use their hands well, then they get a lot of stuff done sometimes.
Yes. So Andrew kind of touched on this a little bit, but when in his answer there a minute ago, but what are there avenues available for interns to continue their research with you after their summer program has ended? Are there ways for them to kind of continue their research with you if they were interested?
Andrew, you go, because it's easiest probably to answer for you.
Sure, I'll say that absolutely. Computational work is really amenable to remote work. This is something that pandemic has really taught us, and so to the point where now my lab, we have full-time employees that live in Massachusetts and North Carolina and Oregon and Texas, and so we're literally a distributed group. So the ability for people to contribute after their summer internship is over is absolutely a possibility. It just comes down to essentially scoping the right project where it's, you can work on it when you have time to work on it, and it doesn't hold back other people's projects and things like that.
That's more challenging for us because our work is based in the laboratory. So if you are coming into the lab over the summer and then you have to go back to school, I have done this before in the past where we had a summer intern come back during the school year. Student was a senior, so scheduling was a little bit more flexible, and we accommodated this person for about 20 hours per week. They were able to continue their work, but it can become challenging when you have school, which should be your primary commitment.
So, I mean, I think maybe globally speaking, the answer to this is it depends on the lab, first of all. So some labs that have computational components, for those it's true. There is also a small feature that is not super well known about the NSF grant, which is that you can apply for extra funds for one person to continue working in the lab. So if, say you live in South Florida, maybe you go to school, I don't know, in the Miami area or something like this. And if you could arrange your schedule in a way that's similar to what Dr. Wang just described, then I think there are ways of doing this. But again, it is challenged by the location issue. So the answer is to be decided on a case-by-case basis.
Great. Let's see here. Kind of shifting gears. What steps do you take to promote a healthy lab culture, and what are some of the indicators of a good and healthy lab culture, in your opinions?
Do you
want to go first again, Andrew?
I am happy to go first. I'm really fortunate I really love all the people we were able to recruit. I think lab culture is critical for attracting people to science and keeping them in science. I think that comes a lot from valuing people for who they are and just building a team that just has diverse backgrounds and expertises, and people who are willing to help each other. So I don't quite know the recipe for building that from scratch, but I will say that a little bit of it is luck, and when you have good lab culture, I think it is it just makes science more productive and more fun.
I think Andrew's touched on my key terms already. It should be a healthy lab culture is fun, and it should be productive. The fun part is kind of easy. You walk in and like there's chatter, people are joking, you know, but at the end of the day, you guys are coming to a lab to be able to do science and perform work. And so we measure that output by your skill sets. What have you learned? What's the new insight? What's the new science that you've generated? So these are my metrics. I gauge that. Partially, the fun is easy to kind of pick up on as a vibe in the lab, but I also informally take an anonymous lab survey for my lab so they can give me candid feedback about how I'm performing as their mentor. What are their needs and if I'm addressing them or not, what are ways in which we could improve that? I take that opinions and feedback really seriously, and I try to correct my behavior based on them.
Yeah, I do something similar, although I guess I haven't done it anonymously. I speak to my people. I meet with them once a year to kind of talk broadly about where they're going. And part of this is also actually a critical feedback for me. And the students have provided me with critical feedback. I make it very clear that this is helpful for me. But I think there's also, obviously every lab does the obvious things, right? Like we have lab parties, the students hang out together, they go, they're friends with each other. And I think, also you obviously want to reward the right behavior and, and not encourage bad behavior. So the students don't compete with each other in my lab. They have different projects that are clearly separated. And if sometimes one person, if one person has a piece of data that can become relevant for someone else's, then we include them as authors easily, just so that no one feels kind of advantaged or disadvantaged to each other, so that they can just help each other, right? And that's, I don't know. It's just the students. That's the answer. But like Andrew said, it's the students.
Great. We have time for probably one more, and I think this is one that will probably be great to leave on, is what advice would you give prospective interns or students that are looking for opportunities such as internships or even grad school? What's some advice that you would give?
I think the advice for grad school is, I think the most important thing is that you find actually a good lab fit. People always think that the most important thing is that, that this is exactly the science that they have envisioned. And I think that's important because you have to be excited about it. But I think it is probably more critical that you are a match for the advisor and the lab in order to be really happy and successful. And that's, I think, what you want to consider as you're testing out different labs. And then I also think you want to think about, you want to do your research, right? Have the previous students published? Where are they going afterwards? Do they get the jobs that they want to get? So those are all, these are not hard to figure out, right? You just talk to the students that are currently in the lab. You can look at the publication record. Those are all pieces of data that are readily amenable, and I think you should think about them as you're deciding a lab that you want to join. For internships, I think the most important thing is just that you get skills, and you shouldn't, they're not forever. You shouldn't worry so much about the science. You should just focus on learning.
Nothing to add.
Yeah, maybe the only thing I would say is that I agree with what Katrine said, is that the the science perhaps is secondary, right? Because you're going to learn stuff in almost any lab that you're going to go into. And so the key is about making sure there's a good structure and for learning. The only other thing I would add on top of that is to say that if you do find a lab you really love, right, I think there is value in essentially carrying that further, right, for as long as you can. If it's, you know, again, 2 consecutive summers or research during the school year, because as Katrine mentioned earlier, right, 10 weeks feels like a long time, but it's not in terms of scientific sort of depth. And getting depth in a project, I think, is really valuable if you want to go to grad school.
Thank you. Thank you so much. I think we are ready to wrap up and move on to our next session. So I want to say thank you so much to Katrine, Andrew, and Mia for all your expertise and advice. Of course, if there's still questions that anyone has, please feel free to enter them in the Q&A, or you can contact us. I'm going to turn it over to Paolo now, who will be leading a intern panel where they can answer questions from an intern, former or current intern's perspective. So thank you again to our mentors.